Okay, here we go with what's called a dihybrid cross. Dihybrid cross. We've already seen that in a monohybrid cross we can track the occurrence of the alleles of a particular trait. One of them will be dominant, one is recessive, and we can make a Punnett square and figure out the probabilities of what those combinations of alleles would be, and hence what the offspring would look like, their phenotypes. Now what we're going to do is we're going to track the probabilities of two traits at once. Two traits at once. Dihybrid cross. And it's really the same idea, it's just a little more complex because we've got to keep track of two different things rather than one. So, the two traits that we're going to track, we're going to use the pea plants again that Gregor Mendel used, because a lot of his genetic laws and rules came from the fact that he actually bred these plants in his garden, remember, and he counted the numbers, and he looked at the ratios, and the ratios match what the Punnett squares were predicting. And so that's how he knew that he was onto something. Okay, so here are the traits we're going to track. We've already seen that a pea can be yellow, or it can be green. That's the color. And we saw that yellow was dominant over green. So we use a capital Y and a small y for the alleles. The dominant Y for yellow and the recessive is the small y, which means green. Okay. Another trait that occurs in peas has to do with the way the seed actually looks, the pea itself. You might have noticed this if you eat your vegetables. Sometimes peas are round and smooth, but sometimes they're all kind of wrinkly. And this is caused by a gene as well. The gene for a round pea is dominant, so we'll give it a capital R allele. And the gene for a wrinkled pea is recessive, so we'll give it a small r. So those are the two traits we're going to track. Now let's look at the genotypes. Those are the phenotypes, yellow and green, round and wrinkled. So what we're going to do is we're going to set up a cross between a yellow and round true breeding pea, and we're going to cross that with a green and wrinkled. That says wrinkled, true breeding pea. If I could spell, true breeding. There we go. So what kind of genotypes does this tell us we have? Well, let's look at the yellow and round one first. The yellow and round is going to be, it's going to have some dominant alleles, right? So the easiest one would be dominant yellow. So that combination could be yellow. And because it's true breeding, we know it has to be homozygous. The other combination for yellow could be this, remember, because the dominant Y could mask the recessive Y and it would still turn out yellow. But that would not be a true breeding yellow, because that little baby Y there, being a green allele, would show up in some of the babies and it wouldn't truly breed only yellow all the time. So we know it's not the case. We know it has to be the actual homozygous, two capital Ys. Okay. Now, what about the genotype for the round part? Well, for the round, we just write beside it the two capital Rs. Again, because it's true breeding, we know that it can't have the small r. This could also produce a round P, but when you cross it, that little baby r is going to show up in some of the babies, and it would not be true breeding. It wouldn't always be round, right? True breeding means homozygous. That's important to remember because it helps when you're figuring stuff out. Okay. Now, what about the eraser's on. What about the green wrinkled pea? Again, it's true breeding, which means it has to be homozygous. It can't carry any other alleles. So that means green has to be homozygous recessive, and so does wrinkled. So there's the genotype for that bee. Now, how do we cross them? Well, we're going to do a Punnett square, but it's going to be a little different because we have so many things to keep track of. The first thing we have to do is we have to determine the gametes that can be produced. And this is the part that you have to remember. You got to remember what meiosis is all about. We can consider that these two alleles are a particular gene. In fact, they're two versions of genes on two different chromosomes that are homologous. Same here, right? And then the other one has a second set of alleles, which is a whole different gene. So here's what this might mean. If we draw a chromosome, during meiosis, that chromosome gets copied. Okay, so there's two versions of it. But that chromosome also has another partner right here that does the same job. Remember that, they're homologous. And so let's say this gene for for yellow, let's go with yellow first. Let's say the yellow gene happens to be a little piece right here on the chromosome. That means there's the capital Y. That means there's also going to be an identical piece with a capital Y here because it's the homologous chromosome. So each of these Ys in the genotype up here represent the alleles here and here. Now, when this process of meiosis happens, these two chromosomes are going to line up in the middle of the cell. Remember the picture we drew? One's going to be here, right? And the other one's going to be beside it. And in meiosis, what's going to happen is the spindle fibers are going to pull this one this way and this one that way. So they're going to be separated. So a capital Y here and here is going to go to either end of the cell, which means that when we're done, we get two separated Ys. So that's how those Ys are going to separate. But look at, no matter how you slice it, these gametes, these are the gametes, these are the sperm and egg cells that are formed. They're always going to have capital Ys. How else can you get anything else? Because that's all you have are capital Ys when you pull them apart. So, understanding that process, we can apply that to the two capital Ys. There will also be in the same cell another set of chromosomes that are homologous, and they will have a gene on them right here, let's say, which is the capital R. They're a different chromosome, different gene for the different trait, but they also will get pulled away. So that in these gametes, not only will you get the capital Y, you'll also get the capital R, which is a different chromosome that's going to be in there as well. See, so there's this one right here, and this one has those two right there. So you have two different letters. Now you have to be careful about confusion. We draw these things together, but this is why we always use the same letter, capital and small, for one gene. This means two alleles of the same trait, whereas this means one allele each of two different chromosomes, two different things altogether. You see why that's important? The R and the Y are not homologous. They have nothing to do with each other. In fact, Mendel formed a law called the law of independent assortment, that means that these genes could go either way. Because when they line up, think about it. In this case, it won't matter because the alleles are the same. But when they line up, one goes one way, one goes the other. But what if they had lined up upside down? Then this one would end up going to the top and this one would go to the bottom. It doesn't matter. There's no law or rule that says it has to line up upside down or not. With two of the same alleles, it doesn't matter. But what if you had a chromosome that had this combination? What that would mean is that on the first chromosome, right here, and its homologous little partner, right here you'd have a capital Y, but on the same spot on the homologue, you'd have a small y. Which means when they separate, the small y goes one way, the big Y goes the other. And then you could see how it matters. But of course, that little Y then could end up with a capital R from another chromosome or whatever. So there's lots of combinations with these dihybrid crosses. okay? And we just have to kind of think this through. Now, it's actually quite simple. I'm drawing all these pictures because I want to remind you about the meiosis process, because that's what causes it all to happen. Mendel didn't know any of that. These pictures Mendel would have thought were crazy. All he knew is that somehow a Y and an R get separated and they go together, okay? So let's go back and erase that, and let's draw the possible gametes that we could get without doing all the meiosis stuff here. Let me just erase all my pictures that I was showing you. Okay? Okay. So, meiosis means that the two Ys are going to separate. So we can do it like this. We can just separate them. I'll draw some little arrows just for fun. We're going to get a Y and a Y. Okay, that's all you do, separate the Y's. Now we also have to separate the R's. One of these R's will go this way with this gamete. So we'll draw it like this. And the other R will end up over with this one in the same meiosis split. So these are the possible gametes. Well, they're both the same. They're both the same. So the only possible sperm or egg we can produce with this combination is this one. That's the only possible gamete you can get, the only possible combination. You're always going to get a dominant yellow allele and a dominant round allele in every sperm and every egg. By the same token, we can look at the other one. Let me just continue this line down here. We can go back up to the yellow one, and we can see that one of these green, these are babies alleles, so they are recessive, will go to one sperm and the other will go to the other. And of course, then we can split the R's too. This R will come over here, and this R will come over here in that same division. That's the other chromosome being pulled apart. So these are the gametes you would get here. Well, there's only one, right? They're both the same. So the only combination we can get is this. So this would be an easy cross. With only one gamete, the Punnett square has only two spots. We're going to take this gamete and put it here. Don't do it like this, because you'll be confused. Don't put the R and the Y and the R in a separate box, because we did that when we had the separate alleles of Y. But here, this Y and R are in the same sperm, aren't they? Or the same egg. They're two different chromosomes. We're keeping track of two chromosomes instead of one. So they go in the same gamete, they go in the same box on the Punnett square. Keep that in mind, okay? Over here, this is the other gamete. The only possible cross, when we put them together, is a big Y, little y, and a big R, little r. So what does this mean? It means that 100% of the offspring, the babies, will have the genotype heterozygous yellow and heterozygous round. They will still look yellow and round because the dominant gene masks the other one for both traits. And that's why, remember, they were true breeding, right? So they have to always produce yellow and round. But now we're going to do something different. This is the F1 generation that we just did. We're going to take some of these baby peas that we just made, some of these guys, and we're going to cross them and see what happens. So we're going to take a second cross based on the F1 generation and try to cross this. We're going to take one of these and cross it with another one and see what we get. How do we do that? We have to think about the possible gametes. Okay. Now, because these are going to separate, let me just draw the two genes again. We got to do the Y's and we got to separate the R's. Okay? Now you're gonna shortcut this eventually. You're not gonna do it like this every time. I'm just doing it like this to help you see the process, right? You're gonna be able, some of you will be jumping on this right away and say, oh, I can just do it in my head. Here's what we get. We're going to get two possible things. We're going to get a Y over here and a separate Y over there, right? And the same is going to happen for this guy. So because they're both the same, I don't need to do it twice, do I? I can just do this side, and we know the other side will be the same. It's going to also have a y and a y. Well, you know what? I'm just going to do it anyway. Why not? It's not like we're in a big hurry to go anywhere. We're all isolated at home, and we've nothing better to do than watch videos about dihybrid crosses. Thank you, coronavirus. All right. Now, here's the thing. The R's have to separate too. But they could separate in two different ways, couldn't they? We could have a big R going with the big Y over here, and the little r going with the little y. But remember, in meiosis, what if that chromosome was situated the other way? What we might also get is the little r going with the big Y and the big R going with the little y. So that big R could go to two different places, right? Oops, that's not right. Hang on a second. That big R could go here, but it could also join up with the other R. So the one that went this way, maybe instead it goes down here and you get this combination. And maybe instead this little r went with this guy. So basically what you have to do is you have to take the first gene and split it up, and then look at the other gene and realize that the big R could go here or the big R could go here, and the little r could go with this one or the little r could go with that one. So what do we have here? We have 4 different types of sperm. We have this possible genotype, this possible genotype, this possible genotype, and this possible genotype. 4 different sperms can be made when you split up these 4 alleles. And the same would be true on the other side, right? So if we were to think that through, this capital R could go here, but it could also go over here. There's no way to know which way it'll go. And the capital or the small r could do the same thing. The small r could go over here. Whoops, those stupid pens. The small r goes this way. Still having trouble here. Okay. Or it could go this way. Small r. Oh, sorry. Blue. If I had a piece of chalk in my hand, I wouldn't have to make decisions about all these colors and pens and things, and then I wouldn't get all confused like this. So you get the same thing, right? You get this guy, this guy, this guy. Oh, we didn't draw him there. Which one was that? That was the capital R. And this guy. So this guy also produces 4 different four different sperms or eggs. So now to do the cross, we have to cross all 4 with all 4. So our Punnett square is going to be huge. We're going to have to make a Punnett square that has room for 4 gametes, all 4 on one side and all 4 on the other. So we need a big one. So I need room for 4 on this side and 4 on this side. So let's take the gametes from this guy first. Let's take those 4 gametes and let's write them in the places on the other side. I'll write them well, I'll just do them in black because that's how you're going to do them anyway, right? Oh, what did I just do there? Okay, let's write them in black. So what do we have? We have this possible gamete right there. Got him. We have this possible gamete. And the order doesn't really matter. Just as long as you got all 4 in there. This possible sperm or egg, and the last one, the little baby one. 2 recessives. And of course we have the same thing on the other side. Now in a different cross you might have different gametes on this side. So whatever these gametes are, you put them on the other side. So you put one here, got that one. You put little y, big R here. Doesn't matter what order, as long as you get them all in there. And little y, little r. All right, so you see how big the Punnett square is. Now we do the cross. Okay, so I'll change the colors just so we can see the cross. I'll do them in blue. So here's what we have when we cross the first one. 2 capital Y's and 2 capital R's. Go across the top. Capital Y, small y, 2 capital R's. Capital Y, capital R, small r. Okay? And in this one, capital Y, capital R, small r. You usually write the small r first. It wouldn't be wrong for this one that I just wrote to write it like this, let's say. But that's just kind of like backwards, and we just don't do it that way. So within each set of genes, or each set of alleles, write the capital first and the small second. It's just kind of the way it's done. Okay, so here we have a situation. We have a small y and a big Y. So I'm going to write a big Y first like that. Big R, big R. Okay, the next one is 2 small y's and 2 capital R's. Then we have a big Y, a little y, big R, a little r. 2 small y's, a big R, and a little r. All these combinations could happen. Big Y, big R, little r. You'll notice that some of these combinations, though, are repeated. So we'll talk about that in a minute. Big Y, big R, little r. Big Y, 2 little r's. Big Y, 2 little r's. And then lastly, big Y, big R, little r. Little y, big R, little r. Big Y, 2 little r's. And then we have this little guy. Okay. Now, let's see if any of these combinations are the same. Let's do the genotypic ratios. So let's start with, I'll do them in red underneath here. Let's start with this combination. Well, I see it right here. So I'll cross it out there. But I don't see that combination anywhere else. There's no other place where I have 2 capital Y's and 2 capital R's. Now count our boxes. Count all the blue boxes. There's 16, right? So this is a 1 in 16 chance or probability. 1 in 16. It's very small. If you think about your decimals, a quarter is 0.25. An eighth is half of a quarter, 0.125. So a 16th is half of an eighth, which would be 0.055-ish or something like that. So that's about a 5%, 5 or 6%. You can do it on your calculator. I don't have mine handy, but actually it'd be 0.0625. That's what it would be, sorry. Let me just do that properly for you. Let's do the math right. 0.0625. So it'd be about 6%. So that's approximately 6% of the offspring would have that genotype. Okay, let's do the next one. Let's look at the next combination. I'm just going to go across the boxes. Let's do this one. Capital Y, small y. Capital R, small r. Let's do that one right there. Okay, well, whoops. Capital Y, small y. Capital R, capital R. Are there any other ones like that? And there is. There's one here as well. And then I don't see any others because you need 2 capital R's and 1 capital Y. So that means that 2 out of the 16 boxes will be this genotype, which is double. So that's about 12%, isn't it? 12 to 13% if you round it off. Okay, let's do the next possible combination. Let's do this one. So I'm going to cross those other 2 out. We got them done. This is 2 capital Y's and a big and small r. All right. Are there any other combinations like that? There is over here. 2 capital Y's and a big and small r. But that's the only place where I see it. So again, we have 2 spots out of the 16 for this genotype. 2 out of 16 boxes. which is approximately 12% of the babies will have this genotype. Let's do the next possible genotype. Let's go across here to this next one, which is one of each. So it's a Y and a little y and a big R and a little r. Let's go see how many of those we have. There's one right here. There's one right here. There's one right here. And there's one right here. There are 4 places out of 16 where you get this combination. So this is 4 out of 16, which is double 12, which is pretty much 24 or 25% approximately. Okay, and there were no others. So let's just cross those guys off the list. Now we've got that. Let's go to the next one. Let's just kind of keep going down the chart. Here's one right in the middle here that looks like, let's do him now before we forget about him, because he's going to get buried in red there and we might miss him. So 2 small y's and 2 capital R's. How many of those are there? 2 small y's and 2 capital R's. I only see 1. That's the only one. So we have a 1 out of 16. And remember, that's about, what did we say? That was about 6%, give or take, approximately 6%. You can just leave these as fractions. You don't have to write them as percents. You can write 1 out of 16. That's a valid number. But some people just hate fractions, so I'm doing the percents as well. But if you're doing an assignment and you just leave it as 1 out of 16, that's fine. That's fine. You don't have to necessarily reduce all of them or do any of the math. Okay, the next one, 2 small y's, big R, small r. 2 small y's, big R, small r. How many of those were there? 2 small, I see another one right over here. And that's the only 2 that I see. So 2 out of 16, right? And that's approximately 12%, give or take. That's about an 1/8. Okay, cross those out, we're done. The next combination, 2 big Y's and 2 little R's. Okay, how many of those? Let's go count them up. I only see 1. 2 big Y's, 2 little R's. I only see 1. So 1 out of 16, which is about 6%. Next combination, we can see we're almost done. Let's do this one. 2 different Y's and 2 little R's. And if you go back to the square, there are 2 out of 16 for that combination. Two out of 16, which is about 12%. And then the last combination, let's cross those ones out. Oh, we forgot to cross this guy out last time, so get rid of him. And then we just did these 2, so get rid of those. Our last one is this one, the double baby. The double baby letters, the double recessive. And there was 1 out of 16, which is approximately 6%. Now, we can make sure we did this right. If we count up 1 and 2, that's 3, plus 1 is 4, plus 2, that makes 6, plus another one makes 7, plus 4 makes 11, plus 2 makes 13, plus 2 makes 15, and 1 makes 16. Because obviously we have all the boxes. We have to make sure we're 16 for 16. So you can double check your fractions and make sure you have to come out even. Now, some Punnett squares will have 16 boxes. Some Punnett squares will have fewer boxes. You might have 12 boxes or 9 boxes or even 2 boxes. As long as the fraction comes out same on top and same on the bottom, you're going to be okay. Now, what are the phenotypes? Well, we can just kind of go back here. I'm going to erase this couple of things here. We can write the phenotypes right in beside. These are the genotypes, right? But, well, actually, we better do it below. Let's write in the phenotypes. Phenotypes. Well, there's 4 possible, I think, phenotypes. Let's see what they are. You can be yellow and round, or you can be yellow and wrinkled. Or you can be green and round, and green and wrinkled. Those are the only 4 combinations. So let's go up here and let's look at them. The first one, 2 dominant, that has to be yellow and round. Has to be. The next one, definitely round, but look at this. There's a dominant yellow still there, hiding the other one, so that's going to look yellow and round. Look at this one. Yellow for sure, and also round, because of that dominant R that's hiding the small r. Look at this one. The dominants will hide the recessives. This will still look yellow and round. Over here, though, we have definitely round, but these 2 small guys mean green, right? There's no yellow allele to dominate. So that's green and round. Down here, oh, green. But this capital R will still make this round. Over here, yellow, but the 2 recessive R's mean wrinkled. And then down here, this will still look yellow because of that dominant Y that's going to mask the small y, but the 2 small R's mean wrinkled. And our last combination means 2 little greens. There's no dominant yellow there, and there's no dominant round, so green and wrinkled. So what you'll notice is, out of the possible combinations, out of the 4 possible phenotypes, we're going to get, well, let's look at all these squares. There were 16 squares, right? So 1, 2, 3, 4 were yellow and round. So 4 out of 16 possibilities were yellow and round. Let's look for yellow and wrinkled. 1, 2, and that's it. Yellow and wrinkled. So yellow and wrinkled is 2 out of 16. Let's look for green and round. 1, 2, and that's it. That's 2 out of 16. And the last one, there's only 1 possible combination for green and wrinkled phenotype, 1 in 16. So these are the phenotype ratios. These ratios up here in red were the genotype ratios. Okay? So what this means is this. If you cross these P's, then you're going to get a 4 in 16 chance. Right? Doesn't mean you have to have 16 P's and exactly 4 of them will be yellow. It just means that every possible seed or every possible combination, whenever you cross these plants, there's a chance of 4 in 16 of getting a yellow round P. 4 out of 16. Now 4 out of 16, that reduces to 2 out of 8 or 1/4, right? 2 out of 8 or 1/4. Four and 16. I'm just going to double check my numbers here. Yellow and round. Oh, you know what? It's not. Is it 14? Yeah, it is 16. So 1, 2, 3, 4. That's 16. Just checking. The reason I'm checking is because I know that we're supposed to get 9 out of 16 for yellow and round. So Let me go back to the Punnett square here. Oh, I know why it's 9. My mistake. Sorry, folks. I got a little carried away. We have to combine this. This is yellow and round, but there was only one of them. See that? This was yellow and round, but there were 2 of them. This is yellow and round, and there were 2 of those. So what we actually have to do is, it's not 4 out of 16. It's 4 and 2 and 2 and 1. So it's 9 out of 16. So that's my mistake. Sorry about that. 9 out of 16. I should have noticed that sooner because I know that there's supposed to be a certain ratio. That's 9 and 16. We got our charts sort of confused, right? If you don't want to do it this way, what you can do is go right on the square and circle the yellow and rounds. Watch. 1, 2, 3, 4, 5, 6, 7, 8, 9. So those guys were all yellow and round because they all had at least one capital Y and one capital R. And there are 9 of them. So that's what we have to do. 9 out of 16. Now let's go back to our wrinkled and yellow and recount that. Wrinkled and yellow, we had 2 here and 1 here. So that's 3 out of 16, right? 3 out of 16. If you do it on the square, wrinkled and yellow means 2 small r's and at least 1 capital R. So wrinkled and yellow, 2 small r's and 1 capital Y, I meant. So 2 small r's and 1 capital Y, right there, and right there, and right there. Those 3. So see, we've already gotten, I'm going to cross out all the ones that we've already taken care of there. So that was 3 and 16. And then the last, the next one, green and round. There are 2 places, but again, look, it's a 2 and a 1, so we have to count that as 3 out of 16 as well. I'm glad I caught that before we went too far. So to get green and round, you have to have 2 small y's for green, but at least 1 capital R for round. So there's 1 up here, 2 small y's for green, and there's 1 over here, and there's 1 over here. So those 3. And then lastly, you'll see, but there's only 1 out of 16 for the green and wrinkled. Sorry about that mistake. So we've got to make sure we get those phenotype ratios. Got to remember that each of the ones here in red, you know, how many were there? Well, there were 2 out of 16. So you got to count that one twice, right? That's 2, 4, and 4, and 1. That's how we got those. Okay, so we got that done. Here are the phenotypic ratios that we expect. And the reason why we know this makes sense is because when Gregor Mendel took these 2 P's and he mated them over and over and over, probably hundreds of times to get the numbers, he saw these particular ratios show up. So what this means is if you mate these 2 plants, there's a 9 in 16 chance that the baby will be yellow and round. There's a 3 in 16 chance that the baby will be yellow and wrinkled. There's a 3 in 16 chance of being green and round, and a 1 in 16 chance of being green and wrinkled. That doesn't mean you have to have 16 pea babies. It just means that every single one has those chances of being those characteristics. Okay? It doesn't matter how many you have. And that's how you do a dihybrid cross. So it's a little more complex. All right? So maybe what we'll do is one more example. Some of you might feel that you've got it, but I'm going to do one more example without making my little mistake there. And I'm going to do just a different situation, and we'll do it rather quickly just to make sure we got it. This time, we're going to take this guy, who is going to be homozygous yellow and heterozygous round. That's his genotype. And we're going to cross it with this guy, who is green and heterozygous round. Okay? All right. So what do? We figure out the gametes that we can make. So we have to split up the Y's. Right? Split up the Y's. And I could get a Y with a big R and a little r, like this. Or they could split the other way, and I could get the little r on this side and the big R on this side, but it really doesn't matter, does it? These 2 are the same, And these other two right here are the same. So the possible gametes, that's all we care about is how many there are possible. The possible gametes here are a big R, big Y, big R, or a big Y, little r. Those are the possible sperm and eggs you can get by doing the combinations. Okay, let's do the other one. If we look at this one, we can get a little y and a little y, right? And now the big R could go this way with this guy and the little r over here, but they could also go the other way. So I could also get two little y's, and I could have the big R going the other way over here and the little r going there. But again, doesn't matter. These two are the same as these two. So there's only two possible gametes that I'm going to get from this one. I'm going to get a little y, big R gamete, or I'm going to get two little guys like that. So those are the gametes I have to work with in my cross. So let's see what would happen. So I do my cross. Now notice this time there's only two of each. Last time there was four of each. So that was a bigger square. This one, I only need two on one side and two on the other. So my square can be a little bit smaller. So I'll put the gametes for the first one over here, and these gametes I'll put up here on top. Okay. Let's do the crosses. Big Y, little y, big R, big R. Big Y, little y, big R, little r. Big Y, little r, big R, little r. And then big Y, little y, two small r's. Okay. Genotype ratios. I have this possible combination, right here, and that's 1 out of 4. This time it's out of 4 because I only have 4 boxes, not 16. The other combination I notice is there's 2 of them. I'm going to circle them in blue. It's this one. They're both the same. So for that combination, which is big Y, big R, little r, I have 2 out of 4. And then the last one is right here, and that one is big Y, little y, and 2 small r's. And that combination is 1 out of 4. So those are the genotypic ratios. 1 out of 4, 2 out of 4, and 1 out of 4. And half the babies are going to be this one. 2 out of 4, that's a half. And then a quarter will be this one, and a quarter will be this one. Probably, probabilistically speaking, right? So if you have 1,000 babies, 500 will be the middle one. Probably. And then 250 will be the first one, and 250 will be this last one. That's what you could expect. It'll never be perfect because probability is not always the same as what actually happens. It's just prediction, right? But it'll be very close to that. Okay, the phenotype ratios. For the phenotype ratios, I have to think about the possibilities here. Now, I can write them all out. I can write out yellow and round. I can write out yellow and wrinkled. I can write green and round and green and wrinkled. But there's no guarantee you're going to get all 4. When I go back up here and I look at this, if you look at the actual Punnett square, every one of those 4 combinations has a capital Y, which means that every one of these babies, 100%, will either, they all have the same. They all have the capital small y. So they'll all be yellow, and there's no chance of a green pea because you need 2 small y's to get a green pea. So guess what? I can cross these right off the bat. I'm either going to get yellow and round or yellow and wrinkled. And if you look at this, all the capital R ones will be round. So there's 3 of them right there in black. Those all have at least 1 capital R. So 3 out of 4 are going to be yellow and round. And if you look up here, here's yellow and round. Here's yellow and round. 1. And there's 3 out of 4 for those 2 yellow and rounds. Because this the R right here masks the baby, so he's still round, even though he has the small r. There's only one combination where I could get wrinkled with the double small allele, the double recessive wrinkled allele. And of course, that's 1 out of 4. So the chances of yellow and wrinkled are 1 out of 4. And there you go. There's the phenotypic ratios, 1 out of 4. And that's how you basically do it. So this one was a little bit simpler because the possible gametes were less. The one I started with was actually the most complicated one, where you have 4 different possible gametes way up here for each one, right? We went through it. Anyway, sometimes it's not that crazy. Sometimes there's only this. But make sure you spend the time to figure out the possible gametes and don't just go throwing letters into the box and go nuts. If you get this part wrong, if you don't get these gametes right here figured out, then your Punnett square is going to be wrong. Okay? And then you're going to have all your wrong numbers. So that's how you do the dihybrid cross. I'm going to post an assignment relatively soon. I'll give you some pages in the textbook that are relevant that you can look up, and hope